DNA stands for deoxyribonucleic acid and is the molecule containing genetic information for all living organisms. It can be seen as the instructions for development, characteristics, and qualities of individuals. DNA is a polymer and is therefore made of monomers, nucleic acids called nucleotides. Each nucleotide is composed of the following: a phosphate group containing phosphate and oxygen, a 5-carbon sugar called deoxyribose, and a nitrogenous base. In DNA, this base can either be adenine, thymine, cytosine, or guanine. C and T are classified as pyrimidines, and A and G are known as purines. Now, in order for these individual nucleotides to become a polymer, they must connect through phosphodiester bonds right here between the hydroxyl of the sugar and the incoming phosphate. As this continues, now we have one strand of DNA. Except, DNA is double-stranded and we need a second strand to complement the first. This second strand will complement the base pairs in that an adenine base on one strand will be paired with thymine, cytosine with guanine. According to Chargaff's rule, there must be equal amounts of A to T and equal amounts of C to G. Now between these complementary base pairs, hydrogen bonds are formed to keep the two strands together. DNA is best described as a double helix, meaning it contains two strands in a spiral wound up. These two strands are antiparallel, meaning one strand goes in the 5' to 3' direction, named after the carbons in the sugar, while the other moves parallel but in the opposite direction, 3' to 5'. DNA replication is the process in which one DNA molecule is copied so that two daughter DNA molecules with the same genetic material can be produced. It is classified as semi-conservative, meaning each daughter molecule will contain one strand from the original parent and another strand newly synthesized to complement that original strand. In prokaryotes, this replication occurs in the nucleoid region of the cytoplasm, and in eukaryotes, it occurs in the nucleus. As for when, DNA replication occurs during the S phase of interphase before cell division, in both meiosis and mitosis, which is when the genetic information is duplicated to prepare for division. To prepare for replication, the enzyme helicase first unzips the DNA so that hydrogen bonds are broken and the strands are separated. To keep them separated, single-stranded binding proteins attach to the strands. Outside of the helicase is topoisomerase, which keeps the DNA from supercoiling or overwinding. This causes the replication bubble, which expands outward on each side. Let's focus on this portion of the bubble. First, primase will make a complementary RNA primer so DNA polymerase III knows where to start. This is because DNA polymerase III can only move in the 5' to 3' direction of the new strand and needs a starting point. For the top, DNA polymerase III has no problem with creating a continuous, antiparallel complementary strand. However, on the bottom, this is an issue. The polymerase can only go to the left in order to go from 5' to 3' of the new strand, yet the helicase continues to move to the right. It's like walking down an ascending escalator. They're in opposite directions, so it must constantly catch up to the moving replication fork. So, the primase must lay down small chunks of RNA again and again. Only then can the DNA polymerase III have starting points while catching up to the helicase. Each of these fragments are called Okazaki fragments. This strand is called the lagging strand, and the one above is called the leading strand. However, there are still chunks of RNA. Thus, DNA polymerase I will come and remove all of those RNA primers and instead replace them with the correct DNA sequences. Now, while it seems done, there are still tiny gaps between the fragments of DNA. To seal those gaps, ligase will go through the lagging strand and glue the fragments together for a perfect DNA molecule. Keep in mind this process of replication will occur on both sides of the replication bubble. As shown when we zoom out, which side is the lagging strand depends on the direction of the fork's motion. At the end of DNA replication, there will be two identical daughter helices, and as said, each daughter helix will have one original strand from the parent DNA and one newly synthesized strand. They will then continue to replicate on their own. This replication is essential for proper cell division and is the blueprint for what genes organisms end up obtaining and passing down to their offspring.